Good day, everyone, and today we're gonna talk about mitosis and meiosis and their differences. Cell cycle is a process inside a cell that helps to grow and divide a cell, which is divided into two major phases. The first phase is the one that helps in the preparation for cell division called the interphase, and the second phase that carries out the division of the cell called the M phase, which has two different processes called mitosis and meiosis. Mitosis and meiosis are both processes in cell division, but they are exactly dissimilar to each other. Mitosis occurs in somatic cell or body cells, while meiosis occurs in reproductive cells, which therefore mitosis give rise to somatic cells which has similar and exact copies from the parent cells. On the contrary, meiosis occurs in males and females that give rise to haploid sperms and egg cells respectively. So let's gonna talk about first what is mitosis. Okay, so mitosis produces two identical daughter cells, and the significance of this mitosis is for the growth and development or the purpose, and also repair and replaces damaged cells. So now let's discuss about the stages of mitosis. So first is the prophase. So the chromatin start to condense into duplicate chromosomes. The nuclear envelope disappears and the mitotic spindle of microtubules developed from the centrosome appears. Okay, then proceed to the metaphase. So the microtubule standing from the opposite centrosome attaches to the kinetochore. So the chromosome aligned to the metaphase plate, and then there is a equal force or tension within it. Okay, so next it will proceed to anaphase. So in anaphase, the microtubules attaches to the kinetochore of the centromeres of each sister chromatids. So it contracts until the connection break. And as you can see in the image, so each member of the sister chromatids moves towards the opposite poles. And the next is the telophase and cytokinesis. Okay, so the telophase. So as the sister chromatids reaches the opposite poles, the chromosomes uncoiled and entangled into chromatin. And then the spindle fiber disappears and the nuclear envelope start to reform. To properly divide the cell into two, cytokinesis takes place. Cytokinesis is the division of cytoplasm. And now let's talk about the meiosis, which is the second type of M phase. So, meiosis is divided into two stages. The first stage is the reduction meiosis, or the M1, or the separation of homologous chromosomes, while the M2 is the equatorial meiosis. Just like the mitosis, it is a separation of the sister chromatids. And now let's proceed to the prophase 1 of the meiosis 1. Okay, so in meiosis 1, okay, the prophase are divided into subunits. Okay, the leptonin, zygotin, pakitin, diplotin, and the akinesis. Okay, so now let's talk about it. So, leptonin is where the chromosome thickens and becomes visible, but the chromatids remain invisible. The centromere begins to move toward to the opposite pole, and then zygotin, where the homologous chromosomes enters synapsis, or the fusion of the homologous chromosomes, or forming the synaptonemal complex. This synaptonemal complex is possible when two homologous chromosomes comes close together. So there must be an association of non-sister homologous chromosomes. And next is the pakitin, or the pakinema. The synapsis is complete all throughout each pair of homologous chromosomes, or there is a crossing over there. So the genetic exchange between non-sister chromatids of a homologous pairs occurs. Okay, the next is the diplonin or diplonema. So the synaptonemal complex is dissolves. The tetrad or four chromatids are visible, and the crossing over points appears as chiasmata, or this is the holding of non-sister chromatids together. Then the meiotic arrest may occur at the same time in many species. Meiotic arrest means the cell divisions will stop in this stage. Okay, until it will be, if there is no meiotic arrest, it will proceed to diakinesis. Okay, diakinesis is when the chromatid thicken and shorten at the end of the prophase 1, the nuclear envelope breakdowns, and the spindle fibers begins to form. And now, it will proceed to metaphase 1. So in metaphase 1, as you can see in the image, so the tetrads line up along the metaphase plate. So that metaphase plate is a imaginary plate. So the chromosome of the homologous pair attaches to the fiber from the opposite pole. So the sister chromatids attaches to the fiber from the same pole. Okay, and now it will proceed to the anaphase 1. So the homologous chromosomes move to the opposite poles as the microtubules contracts or shortened. The sister centromeres remains connected to each, and the chiasmata dissolve. So in the anaphase 1, the chiasmata will be dissolved. And note that there is a reduction of the number of chromosomes moving towards the opposite pole at this point. Okay, and it will proceed to telophase. So the telophase, so in each complement of the homologous pairs reaches the opposite poles, so the nuclear envelope reforms. So the cytokinesis separates the daughter cells, and the end product of meiosis 1 are the two daughter cells which half the number of the chromosomes and with respect to the origin cells, and each consisting of sister chromatids. Okay, and it will be now proceed to meiosis 2. And meiosis 2 is composed another of, again, of 5 stages. So as meiosis 2 is also known as equatorial meiosis, or the separation of sister chromatids, meiosis 2 is a similar to mitosis. Okay. So in prophase 2, the chromosome condense, the centrioles moves towards the opposite poles, and the nuclear envelope breaks down at the end of this phase. Okay, and then it will be proceed to the metaphase 2. So the centromeres of each of the sister chromatids attaches to the spindle fiber from the opposite poles. Each chromosomes align at the metaphase plate, and then it will proceed to anaphase 2. So the sister chromatids, or the sister centromeres, detach from each other, and the sister chromatids move to the opposite poles. And then after that, it will be proceed to Telophase 2 and cytokinesis. So in Telophase 2, it is when the each individual cell sisters chromatids begin to uncoil and the nuclear envelope reform once again, and the cytokinesis proceeds, producing 4 unique daughter cells. So those are the M phase of the cell cycle. And now let's talk about the differences of mitosis and meiosis. Okay, so now in comparing mitosis and meiosis. So meiosis differs from mitosis. So the first two cell divisions occur in meiosis. Only one happens in mitosis. So in meiosis, homologous chromosomes pair off in the first division. So during tetrad formation, two chromatids from a homologous pair may exchange genetic material, or also known as the crossing over. So thus, the possibility of a change in a genetic material of the offspring is great. And on the other hand, no pairing of chromosomes occurs in mitosis. The chromosomes are independent of each other, thus the cells are diploid. They have all the characteristics of the parent cell. So the product of mitosis is a 2 diploid cells, and while the meiosis have 4 diploid cells. Okay, and now let's talk about one of the eukaryotic life cycle, which is the gamethic life cycle. So this gametic life cycle is common in animals and some protists, and the product of this are gametes. So as you can see in the image, so from the diploid individual, it undergoes into reproductive cells, which is undergo meiosis. So after meiosis, it will have a product of a 2 haploid cells. So these 2 haploid cells will undergo fertilization, and after the fertilization occurs, it will create an zygote, which is once again back into a diploid individual, and the cycle will repeat and repeat all over again. So that would be all, and thank you.